we will start with our first talk. And we really wanted to kick off the day with everybody being grounded in knowledge. As you all know, I feel very strongly that knowledge is our power. But if we don't understand what the people on this stage are telling you, one, we're wasting your time, and you being here away from your family and getting caregiver support is honestly a privilege for us. We do not want to waste 1 minute of your time being here. So we need to ground you in knowledge, which means that we're going to talk about different things this morning. What does in vitro mean? What does that mean, and why is it important to develop drugs for our children? We're going to follow that by what does in vivo mean? So in vitro, in a dish, in vivo, inside a body. And then we're going to talk about the UBE3A gene. What is the function of UBE3A? I can tell you what it doesn't do. Meet my daughter. But what we're going to learn is what it is supposed to do. And that's very important for us to understand if we give it back, what symptoms we can address potentially with that. And then we're going to talk about what is gene therapy. Gene therapy is a word we use all the time, but I guarantee you 90% of you don't actually know what it is. So we're going to talk about what it is, really get a good understanding to make very important decisions about your child in the future. You need to understand what we're talking about. So with that, let's kick off the morning with Dr. Michael Boland. Dr. Michael Boland comes to us from the University of Pennsylvania Perelman School of Medicine. I'm going to let him introduce his incredible academic accolades because he's one of the smartest people that I know. He's a neuroscientist who's incredibly brilliant. But the most important title is he's a dad. And I'm going to let him tell his story. Michael.
Thank you, Allison. It's an incredible honor to be here, and I'd like to thank the organizers and FAST for putting this on and for everyone showing up to listen to this. So, let me get my slides up. There we go. So as Allison alluded to, I'm the Strategic Director for the Translational and Clinical Research at the Center for Epilepsy and Neurodevelopmental Disorders, which spans both Penn Medicine as well as Children's Hospital of Philadelphia. But before that, I was a faculty member at the at the Columbia University Medical Center, where I was studying pediatric epilepsies. My background is in developmental neurobiology, pediatric epilepsy, genetics, transcriptomics. And I was using human pluripotent stem cell models, which we'll get into in a minute, as well as genetic mouse models to study a number of monogenic single gene disorders that cause pediatric epilepsy and autism spectrum disorder. Worked on a number of different developmental and epileptic encephalopathies, and it was actually during this time that my son Lucas was born with, strangely enough, a developmental and epileptic encephalopathy. He was having seizures the day after he was born, and I had to convince the neonatologist on call at the hospital that he was having seizures. And finally, they agreed with me, and on the second day of life, he was admitted to the NICU. Every test came back negative, MRI, spinal tap to check for viral infection in the central nervous system. Everything came back except for abnormal EEG. And then a couple weeks later, we got the diagnosis that he had a frame shift in a gene called STXBP1. We'll talk a little bit more about STXBP1, and I want to use this as kind of a model system for, as I walk you through how we can develop or use in vitro tools to develop disease models that can help predict pathogenic phenotypes as well as response to therapies. So I want to first walk you through some nomenclature, common nomenclature that I think will be useful for you to hear throughout the day, some genetics, description of non-neuronal cell lines and what they're useful for, and then we'll go into human-induced pluripotent stem cells, genomic engineering, a bunch of different methods to study essentially neurons in a dish. And then I'll give you a real-world application revolved around STX-BP1 modeling in a dish and a gene-targeted therapy to correct some of the deficits seen in those cells. So I just want to give you a quick reminder. Most of you probably don't know what STX-BP1 is. All of you know what UBE3A is, but STX-BP1 is expressed in all neurons, and it's absolutely critical for how neurons communicate. Neurons communicate through neurotransmission mediated by synaptic vesicle release that goes from one neuron to the next neuron, and thereby initiating the action potential and electrical propagation. And so STX-BP1 is critical for release of those synaptic vesicles, exocytosis and neurotransmitter release into the synaptic cleft. So it's absolutely critical for communication in the brain. So, as Allison said in the opening, there's a couple of different ways that we can develop disease models. One is in vitro, and this is in a most likely a cell line in a dish, and we can manipulate those cells in a number of different ways. And then there's also in vivo. Rodney is going to talk to you after this about most likely about mouse models of different diseases. He's really terrific at characterizing behavior and whatnot of mouse models of disease. But we're going to focus a little bit on in vitro for the next 15 minutes or so. And so in vitro means just in a dish. We can take cells such as cells treated with a drug, Angelman syndrome, neurons, what have you, and study DNA, mRNA, the UBE3A protein. That's the structure of it down there at the bottom, that squiggly thing. There was a Nobel Prize awarded last year for AlphaFold, which is an AI method to predict protein structure, which is going to revolutionize drug discovery. And like I said, there are a number of things that we can do with these cell models. And for instance, drug screening, treating them with drugs, different antisense oligonucleotides. We can look at cellular morphology through high-content imaging and also look at metabolic profiling to see how the metabolism of neurons or cell types affected by disorder are changed and are different from control cell lines. So going way back to the basics, you know that you have two sets of chromosomes, one set from your mother, one set from your father. Each one of those gametes had 23 chromosomes, and when they combine to form the embryo, now you have 46 chromosomes, and that's called diploid. Another technical term, scientific term that you'll probably hear is transfection. This is moving DNA into a cell using a non-viral means like a lipid nanoparticle, which you'll probably also hear about today. And there's also transduction, and this is the process by which viruses enter a cell to deliver a cargo such as mRNA or DNA. So some of the more common cell lines that people use are actually pretty terrible, because if you look at each one of these cell lines, whether it's the human embryonic kidney, the epithelial cancer, HeLa, the retinal pigmented epithelium, or the neuroblastoma cell line that I've indicated here, look at the number of chromosomes here. They're highly aneuploid. These are essentially cancer in a dish. And so these don't represent very good cell models of disease, but they do have their utility because they're very easy to work with, they're easily transfectable, and you can use them in really high throughput to screen tens of thousands of drugs. This is one of the first steps you would use, a cell line such as this, to give you an indication that, for instance, if you're screening an antisense oligonucleotide or some small molecule or even virally delivered genes, whether or not this is going to have an effect in a real disease model. But these, like I said, these aren't typically used for disease modeling because they're incredibly artificial. In 2007, Shinya Yamanaka published a paper where they took adult fibroblasts, so just a fibroblast taken from a skin punch biopsy, grown out in culture, and they were able to revert that cell type from an adult person back into what resembled a human embryonic stem cell. That's a stem cell, it's a fleeting cell fate. Very early in development, these embryonic stem cells give rise to every cell type in the body, the entire embryo proper. He won the Nobel Prize for this discovery in 2012, and this became the de facto way to start to generate and look at pluripotent stem cells, because you didn't have to destroy an embryo to derive them. You could take dermal fibroblasts or mononucleated cells from the blood and apply these viruses that contain these reprogramming factors and turn these back into what's called a human-induced pluripotent stem cell, which is also able to give rise to all cell types in an adult organism. And you can take these cells and using small molecules and other proteins and whatnot that mimic the developmental cascades that give rise to different cell types and germ layers, you can generate a number of different cell types: muscle, heart, pancreas, what have you. We can also generate all the cell types in the brain, for instance, neurons or astrocytes. And these pluripotent stem cells, these iPS cells, were proven to be biochemically equivalent to embryonic stem cells when a group that I was involved with at Scripps Research were able to clone mice using these cells. So demonstrating that they truly can give rise to any cell type in the body, because we cloned mice that were fertile and could breed and whatnot. So there are a few different ways to make these iPS cells. All involve that reprogramming using those reprogramming factors, but there are some different models that we should consider. So this is a trio or a familial model where most of you have had whole exome sequencing done, and when they do that, if they're looking for a de novo mutation, they take your genomic DNA, your spouse's genomic DNA, and your child's genomic DNA, and they sequence it, and that's called a trio. And then they use the information there to see what's different between your child and you for de novo mutations. So in this case, we've derived iPS cells from a mother, a father, and an infant with a neurological disorder. So the iPS cells derived from each of these people represent essentially them in a dish. So the iPS cells from the child with a neurological disorder will differentiate into neurons that harbor the genetic defects and physiological defects pertinent to that mutation. Another way to use these iPS cells is isogenic model, and this just means the same gene. Isogenic means the same genome. And so the way to do this is you take you generate iPS cells from an infant with a neurological disorder, or any disorder for that matter, and then you can use genomic engineering, such as CRISPR-Cas9, to correct that mutation. And now you have a corrected isogenic control line that you can compare to the mutant line. And theoretically, the only difference between these two is the mutation that's found in the proband. Similarly, we can also take an unaffected iPS cell line, say an iPS cell line generated from me or you, and use genomic engineering, but this time to knock different mutations into that cell line so that we can study the effect of different mutations of the same gene on the same genetic background. This is an incredibly powerful technology because so much of what makes us are driven by genetic modifiers that modify disease phenotype, which is why we have heterogeneity in our patient populations, genetic modifiers. So these iPS cells, as I alluded to, can be differentiated into neurons using small molecules and proteins that mimic developmental morphogen gradients in different regions of the brain. It goes through a timed series of events that go through neural rosettes, which mimic development of the embryonic neural tube into neural stem cells that give rise into progenitors that differentiate into immature neurons, and then you get more mature neuronal networks that in the dish can talk to each other and we can measure their activity. Another way to do this is to forcibly overexpress a gene called a transcription factor that is involved in cell fate decisions. In this case, it's NGN2. And when you express NGN2 in an iPS cell, you skip all of this development and you go basically right to the neurons. So in a dish, this saves you time and money. And then the third way that is probably the most commonly used now, the NGN2 and these organoids or spheroids. The organoids or spheroids are similar to a naturally occurring developmental model in 3 dimensions. Sometimes you'll hear this referred to as mini brains, and that's definitely not the case. These don't have consciousness or anything like that. But it's a 3-dimensional representation of brain development in a dish, and it's incredibly powerful. We can even derive organoids from different regions of the brain. For instance, this CO is a cortical organoid, so the dorsal forebrain, versus the MGEO, which is the medial ganglionic eminence, which is a region in the center of the brain where certain neurons called GABAergic interneurons or inhibitory neurons come from, and that's a huge interplay in the brain, in brain development. So when thinking about which type of model you want to develop for a given disorder, you need to think about the biology of the disorder of the gene that you're studying, and then you go for an appropriate model based on what you want to get out of that. Organoids are tricky. They generally take about 4 months to get good neuronal activity. You can get similar activity from NGN2 cultures in less time. It's just each one has pros and cons, and the scientist will decide which is best for their model. But let's talk about cortical organoids. You can make hundreds, if not thousands, of these in a couple of hours. And they're tiny. They look like tiny little balls of cells the next day. And then they just grow over time into about a little bit smaller than the size of a pea after 4 or 5 months. And when the brain is developing during very early embryonic development, it's built in an inside-out fashion where these very early radial glia cells divide into these intermediate progenitors that then divide into a different type of cell called an outer radial glia that extends this really long arm or process all the way up to the top of the developing brain. And the immature neurons that divide out from these outer radial glia cells crawl up these processes to their specific genetically determined, temporally and spatially determined place in the brain. And newborn neurons stop Other ones crawl past them, stop, and then it's so on and built like that. And we can use different markers of cell types to track which of these, track these cell types. For instance, the deep layer neurons you see have a gene called TBR1 expressed, or CTIP2, whereas the superficial or top layer neurons have BRN2 or SATBP2. And during organoid development, we can actually track to see that this is faithfully recapitulated, and it is. We start with these early neuronal progenitors that then start to give rise to deep layer neurons that then give rise to GABAergic progenitors, excitatory neuron progenitors, and finally, after 3 or 4 months, we get highly synaptically organized neuronal networks within the organoid that hopefully the organoid contains the cell types that we want. But not all organoids are created equal. In the developing brain, in the developing cortex, there are 2 neuronal subtypes that are incredibly important, and their interplay is very important. These are excitatory neurons that mediate most of and these respond to glutamate, the neurotransmitter glutamate. These are the main excitatory output, sensory output from the developing cortex. And then you have the inhibitory neurons, which are there to kind of put the brake on the excitatory neurons and make sure they're not too excitable. There's a lot of things that can go wrong in these interactions, which result in hyperexcitability, which is attributed to epilepsy. This interaction between these cell types is extremely complicated. There was a paper that just came out that in the developing mouse cortex, there are thousands of different types of inhibitory neurons that all have different functions on regulating the activity of excitatory neurons. And so there are a number of different ways to make these organoids using different protocols from different labs. And what I want you to appreciate here is that the developing neocortex has about 25% inhibitory neurons. If you look at some of these protocols here, the inhibitory neurons are indicated by, or the percentage of inhibitory neurons is indicated by the green pie in the pie chart. And you can see that some of these protocols pretty faithfully recapitulate what you see in the developing neocortex, whereas some, like the Beery et al., have hardly any GABAergic interneurons. Well, I'm interested in STXBP1, which regulates how excitatory and inhibitory neurons communicate. So I want to use a protocol that makes an organoid that contains the cell types that I need to study the intricacies of the neuronal network and how the neurons can actually communicate. Again, not all organoids are equal, even using the same protocols. So this is data from my lab at Columbia, where we took iPS cells and their controls, made organoids from different cell lines, and we looked at their morphology, or how they look, and their activity. And you can see that on the left, those are beautiful organoids. Yes, I'm a nerd. That's a beautiful organoid. The ones in the middle, not so good. This is at 18 days. At 18 days, very early in the development, they start to form these blebs that are almost reminiscent of embryonic yolk-like structures. And then they just implode and die. And on the far right, we have nice, beautiful organoids, but when we go to measure their electrical activity, they're largely inert. So there's this trade-off. You want something that has really nice morphology and structure, but also you want something that has a good activity profile, electrical activity profile that you can measure. So let's get into the application in the last few minutes of the talk and see how you can try to put all of these different in vitro tools together to model a disorder and develop a gene-targeted therapy. So obviously, when Lucas was born, I made iPS cells from him. That's what I do. And from me and my wife. My wife's iPS cells were terrible at making organoids, which I like to point out often. Mine were really good, actually. So the first thing we did was we differentiated these iPS cells into just 2D neurons in a dish because I wanted to see if the neurons themselves recapitulated what we thought was going on with Lucas's mutation, which is a frame shift or a protein truncating variant. So he should have haploinsufficiency and about a 50% reduction of STXBP1 protein. And so we did that. We differentiated these in a dish, and after about 65 days, or two months in a dish, we harvested the protein from the neurons, and we checked the expression levels of STXBP1, and as expected, Lucas's neurons contain about 50% less STXBP1 than my neurons. So then we decided, let's go after an organoid model, and I won't go into all of the details and data that we accrued over time developing this organoid model, but I'll show you some of the activity measurements and how they relate to gene therapy development. So this is a blow-up of a single organoid, again, probably about half the size of a pea, at about 4 months old. And this is one of Lucas's organoids. And I'm going to play this movie, and I want you to appreciate the activity within the organoid. So you're going to see a big bright flash, which is a synchronized activity. Synchronous oscillations are common in these closed loop systems like an organoid. And you'll see a big bright flash, and then you'll see some little blips in these highlighted circles. You'll see big bright flashes periodically, and that's these synchronous oscillations. So big bright flash, a little blip. And then you'll see another blip right here. And then more of these synchronous oscillations. So we can actually measure this and start to quantify this, and we could see activity pattern differences between Lucas's organoids and the control organoids. For instance, you can already tell that these bursts in Lucas's organoids are wider, and they have more spikes within the organ, within the burst compared to the control. And as well as these bursts are timed differently. There's more space in between them. They're longer. The bursts are longer. There's more time in between them. So these are all characteristics that we can use to try to modulate with gene therapies. So I'm very interested in therapies that modulate how genes are expressed, whether they be antisense oligonucleotides or using an adaptation of the genetic engineering CRISPR-Cas9 system called CRISPRa. So this is a Cas9 protein that should be able to cut DNA for the editing purposes that I talked about earlier, but the active site of the protein has been mutated, so it can't cut DNA. But you can still hone this protein to anywhere in the genome because it binds these little pieces of RNA that are complementary to different places in the genome. So if you can find these little RNAs, for instance, in the STXBP1 gene regulatory elements like the gene promoter, you can hone this Cas9 protein to that region. Cas9 itself isn't going to do anything, so you need to add what's called a transcriptional activation domain. This brings in the machinery that helps upregulate gene transcription from that promoter. So using this CRISPR activation technology, we can upregulate, or should be able to upregulate STXBP1 mRNA that's then translated into protein correcting haploinsufficiency. And we first screened a number of these different guide RNAs in the shitty cell lines that I told you about earlier, HEC293T and the neuroblastoma cell line, because they're easy, they're quick. You can get a lot of information quickly, and we found that, to our surprise, almost all of our guide RNAs were very effective at upregulating STXBP1 expression. So we took our CRISPR-Cas9, CRISPR-A AAV virus, and we transduced organoids at about 3-month-old organoids with this virus that should be able to correct haploinsufficiency. And after about 2 to 3 weeks, we harvested RNA and DNA and tested whether or not the STXBP1 protein and RNA levels were corrected, and indeed they were. So in Lucas's organoids, we've essentially reestablished the normal level of STXBP1 expression. What does that do to the physiological defects that we identified in the organoid, you know, the flashing movies? Well, some of those little blips that I called your attention to, those low amplitude events we think are microcircuit defects, potentially related to epilepsy phenotypes. And we showed that the CRISPR-A virus could actually reduce the incidence of those low amplitude events, thereby potentially correcting that defect in the organoids. Moreover, if you look at a number of different cells on the y-axis, in this case hundreds of cells, and then over time, what you can get is a picture of activity in the organoid. And so those bursts that I showed you earlier in the colorful diagrams can be depicted by these lines that go up and down on this what's called a raster plot. So you can see in the control, you have very thin bursts in the whole organoid. These are the synchronized oscillations. And in Lucas's organoids, again, the bursts are wider, the dark lines are thicker, and there's a different spacing. They're in general further apart. If we apply the CRISPR-A virus, we can start to correct that. The bursts shorten, the interburst or the time in between bursts begins to shorten, indicating that we're starting to have positive effects on the neurophysiology of these organoids with a gene-targeted therapy. And so this is one very powerful application of in vitro models for developing and testing gene-targeted therapies in a disease-relevant, clinically relevant model. And FAST leaves no stone unturned. They've funded work that is involved in using the CRISPR-Cas9 gene editing function to essentially just chew up the UBE8 antisense transcript. There's another group that used a very clever method to actually turn off the antisense transcript by methylating the region by using epigenetic modifiers that will methylate a certain region that turns off the antisense transcript, thereby upregulating the paternal UBE3A transcript. And then I think just last year, FAST has also doubled down on a CRISPR activation technology similar to what I described to hopefully meet a therapeutic potential for the deletion genotype. With that, I'll stop. Thanks some colleagues from Columbia and the END team at Penn and CHOP, and I'm only slightly past time. Okay.
Thank you so much. So as you can see, that's really complicated. And it's hard to make that simple because it's not. But what I want you to realize is that when everyone's like, why is this taking so long? You need to do all of that, which can take years, before you can ever even put it into an animal to test if it has the potential and the possibility of even working. So our next talk is to talk to us about putting it into an animal to show us the possibility of how it might work, which gets a little bit more simple as we go closer and closer to humans. So I know that was a lot, I know that was complicated, but you can see the elegance and the beauty of the science, and that's why things take a long time and cost a lot of money. One thing I just want to highlight is that I was told the QR code for translations in the brochure is not working, so they're putting a new QR code on every table. So if you need translations, just use the new QR code to get your translations. We want to make sure everybody can hear and understand.